Good morning, good afternoon, good evening, everyone. My name is Dalip Sethi, and I work at Terumo Blood and Cell Technologies. Terumo Blood and Cell Technologies is a medical device company that provides tools and services to advanced therapy providers. Our products, softwares, and services enable customers to collect and prepare blood and cells to treat challenging disease and conditions. Today, I have an opportunity to discuss automation and the flexibility of automation with my colleagues in this panel, industry partners in this panel. I'm really excited. So what I'm going to start with, before I ask my questions, I'm going to ask the panel members to introduce themselves, and I'm going to start with Donna. Donna, if you will, please.
Hi, good morning. I'm Donna Reel. I'm the Chief Technology Officer for Trimvera Immunologics. We have an alternative technology to CAR-Ts called TAC-Ts. And my focus is on process automation and process development and coordinating that with integrating into a proper CDMO environment. And our next participant is Oliver Jari. Oliver?
Good morning, good afternoon, good evening. Olivier Jarry, I'm an investment banker and advisor in New York. We support small and mid-sized companies getting funded, but also making the right choices for their technology and how they integrate with our partners. Thank you. And now Jelena.
Yes, thank you. Yeah, I'm working for the Fraunhofer Institute for Production Technology in Aachen in Germany, and we're an applied research organization with over a decade of experience in lab automation. And our approach towards automation is the development of fully automated laboratories, which are robot assisted. So example for these kind of facilities are the stem cell factory or the AutoStem. And with that, I would like to hand over to my fellow participant, Amina from the UCL.
Hi, my name is Amina. I work as a regulatory manager at our sponsor's office in UCL. We oversee a number of trials, and my main role is the IMP management of the trial, right from the risk assessment, the logistics, licensing, manufacture, and all the way to the sponsor release of the IMP. So we have a number of IMPs that are manually manufactured and semi-automated at the moment. So I can just bring into the table different regulations surrounding that and the submissions and the amendments that happen. And that I'll pass on to Diana, sorry.
Yeah, no problem. My name is Diana. I'm in Zurich, Switzerland. And I'm leading a clinical trial in phase 1 for restoring the sphincter muscle for restoring urinary incontinence. Currently, as I said, in phase 1, doing everything manually with my team and looking for scaling up by introducing automation into the process so that we can reach big enough numbers. Thanks a lot. And next one would be Fabio. You're muted.
Yeah. Good morning, everyone. My name is Fabio D'Agostino. I live in the UK, but I work with a new VC fund which just launched, Claris Ventures, in Turin, northwest of Italy. And the focus is to invest in new high potential therapeutic companies, mainly at preclinical stage, late preclinical stage, particularly in the areas of oncology, immunology, and rare diseases. And will really be some small molecules, peptides across the range, but certainly some cell therapies as well. And before that, I've been working in cell therapy for the past 10 years. And with that, I'll hand it to Joan Hannon.
I think we can move to Tatiana. Joanne is...
Joan is here.
Good morning, good afternoon, good evening to everyone. And my name is Tatiana Golovina, and I'm currently with WuXi Advanced Therapies. I have been in cell therapy for more than 15 years, started in early development in academia at UPenn, then moved to Big Pharma, Novartis, Morris Plains, where we launched the first cell therapy, commercial cell therapy, Kymriah. And currently I'm working with CMO and facing multiple clients and multiple processes and helping our clients to get to clinical and to commercial. And with this, I will pass it to Elvin.
Thank you, Tatiana. Good morning, everyone. And my name is Elvin Vargas. I'm currently the director for engineering and technologies for Fujifilm Diosynth Biotechnologies. I'm located in Texas. Fujifilm is a leading CDMO from early stage commercial manufacturing, actually, and I have been supporting the company in the automation, IT, and project engineering side, being in the industry for 19 years, starting with big pharma, then moving to big system integrators, and now in the CDMO business, supporting all these efforts on gene and cell therapy. And I would like to welcome Kathy Schneider.
Yes, thank you. Thank you for the session as well. So my name is Kathy Schneider. I work with Irby Biosystems and lead their product management and commercial activities. We are an innovative startup company with a microfluidic 2 mil scale bioreactor that we're bringing into the cell therapy market to help them better understand product process as well as media concentrations. And with that, I would like to introduce Stefano.
Thank you, Kathy. Thank you, everyone. Good morning, good afternoon, good evening. My name is Stefano Baila. I'm the Director of Operation of Anemosite, an innovative contract manufacturing organization. Exactly, we position ourselves as a biotech manufacturing organization, and maybe we'll have a chance to explain why later on. But long story short, we work mainly on manufacturing cells with non-viral method for editing them and plasmid manufacturing. Those are the two key factors that characterize us as an entity. And with that, I move to Ramin.
Thank you, Stefano. Hi, everyone. It's Ramin Bagherzadeh here. I'm a global head of business development for cell and gene therapies at AGC Biologics. We are a global cell and gene therapy and biologics contract manufacturing organization with a global footprint across 3 continents. And for cell and gene therapy, so we have a footprint both in cell therapy and viral vectors and plasmid manufacturing. Been in this industry for about 15 years, originally coming from large pharma and Roche, and my most recent role also has been in Lonza, also in cell and gene therapy unit prior to joining AGC Biologics. So very much looking forward to today's discussion. And with this, I will pass it back to Dalip.
Thank you, Ramin, and thanks everyone for this great introduction. And I could not be more happier having this diverse panel from regulatory to product management to commercialization, and having also people, decision makers on the panel. So I'll start with automation and flexibility discussion. So the first question I have, as leaders in cell and gene therapy industry, we all agree that automation of manufacturing technology is key to the future. So when you think of automation in your experience, or when you look at the industry, what is the best time or the development stage to consider automation, and why is that stage important to consider automation? Whoever wants to go first, please.
I think I can go. Well, Donna, go ahead.
I'm not bashful, so I'll go ahead and make some comments. You know, there's a different environment these days than there has been in the past for cell and gene therapy. There is a lot of exciting technology out there that has facilitated automation and has moved from the classical pharma world and biologics to fit to the cell and gene therapy scale. And I think that's hugely important. Both on the vector manufacturing side, the processes are now available to make this a much more commercializable reality. And the same thing is happening with the actual manufacturing of the cell and gene therapy products. That being said, I think it behooves anyone that is in their early stages to aggressively early on look at automation. With that being said, there's a lot of considerations for that. It's a lot of bottlenecks that have occurred in the field with, even if you're it's usually not feasible for a small startup company to build out their own facilities. So you depend heavily on the CDMOs or CMOs. But then you get into scheduling bottlenecks and et cetera there. The more you push to automate your process early on, the more feasible it is for the CDMOs to integrate more and more projects as automation occurs. So I think it's important that as anyone is looking at moving something into phase 1 trials, that they also look at where the process is, hugely understand their characterization and functionality with their existing process, and start aggressively looking into automation and how to advance it, that by the time they start their phase 2 trials, they're automated.
Thank you. Let's go to Elvin. I know Elvin was, and then I can ask Amina, Oliver, and Ramin.
No, I concur with Donna. I think automation is something that you need to think as early as you can, especially when you're building your processes, right? So you need to think about scalability, increased control, recipe-driven efficiency, flexibility, how to minimize errors, especially in operators and the operations when you actually try to repeat the same process again and again and again. Also, in our case in Fujifilm, so we need to think about technology transfer, right? We have multiple locations across the world, and sometimes we need to do tech transfer, how we can make that easier for our partners out there so we can support their processes and their projects, right? So that's key, and that's actually really important for us. And even automation goes, I would say maybe beyond that. I mean, right now we're scaling processes to 2,000 liter bioreactors in single use, or even we're going to 20,000 liters in stainless. So predictive is also something that I think is catching up more eyes, not coming from the big pharma also, but in this new world about the gene therapy. So those are key points that we're currently working at Fujifilm, and I like Donna said, it's something that you need to think soon, and you need to plan ahead, because if not, it can cost more and gonna make it harder for you. So
yeah, thanks, Elvin. Thanks, Donna. So moving to Amina. Amina has raised her hand. One question I would add to that, as we are thinking about automation, we understand early is good, but also, Amina, from your perspective, from a regulatory perspective, what about as early automation and the flexibility, because Elvin touched on the process transfer discussion. So you're muted, Amina.
Sorry. Yes. From a regulatory perspective, I think what the regulators really want to look at is ensuring the safety and efficacy. And of course, the efficacy comes through the clinical trials and actually testing them on patients. So usually what you tend to find is early on in automation and in manufacturing validation, you're focused heavily on the safety. You're focused heavily on what tests you're going and what in-process parameters you're looking at to ensure the safety of the product into the patient. To give you an example, we had a product, a CAR T-cell product, that was manufactured initially manually. So it was all manual specifications. We had the end product specifications, and it worked very well. And we moved it on to the second cohort of semi-automated manufacture. We went on to just get the exact same specifications. All of the validation work was perfect. But however, when we moved on to actually giving it to the patient, we found that the CAR T-cell persistence was much, much lower in the semi-automated process than it was in the manual. So we're having to revert back to the manual process to make sure to see where exactly we've gone wrong and to manufacture there. So I think what we're looking at is making sure that the process is kept very, very simple to actually explore exactly what specifications or what in-process parameters you're looking for, because they can be very, very minor, very, very small, but they will make quite a big difference. So choosing very carefully what your end product specification and what you're looking for is going to be quite paramount in moving on to an automated process, especially if you want to do it early on in the phase of, in your trials.
See a lot of hands, so I'm just going in the order. So Oliver raised the hands next. So I'll go to Oliver and I'll come to Ramin, Zelena, Stefano, and Diana. I know it's a very interesting topic, but I'm picking up on Amina's topic about transferring the process, and automation is key, and also the flexibility and making sure your product doesn't get impacted. So Oliver.
Yeah, just adding to previous answers, actually we finished recently a study with a dozen companies. The first thing was, I think Madonna brought it up, which is really plan in advance. And what we found is in most cases, people are preclinical, they plan for phase one plan for phase two, and they don't see the big picture at once. And this causes a lot of rework, and it's not always easy, but we found out that best practice is actually if you can plan in advance, even if it is very early for the final stage, and almost work it back to make sure that that you don't take a dead end in the beginning with some technologies. The second aspect is devil is not in the detail; it's in the interfaces, and especially if you work with CMOs, CDMOs, all these tech transfers, especially for small companies that are very dependent on their CDMO, are extremely difficult. And when something goes wrong, it goes wrong at the interface. Big pharma typically, or big companies, have a very directive way of pushing this interface and saying what they want. Small companies are the opposite. Sometimes they don't really know what they want, and the CDMO is the one telling them what they want. And if you go back and forth between different CMOs, then it's even worse. And I appreciate the comment of Edwin on this. I mean, these tech transfers are really something to watch for. The third piece, which is more for large companies, is building for flexibility. And we had a number of people telling us that you may design something which is very nice for one thing at a time when you think exactly of that specific process, and then you are stuck because it's not adaptable to something else.
Thanks, Oliver. So I'm going to Ramin and then Zelena.
Sure. I mean, I agree with a lot of points already discussed. So maybe to add, I mean, I think all of us would agree that automation is crucial, but I think the key element is at what point it should be introduced. And I mean, and as we know that the majority of cell and gene therapy players are small biotech companies, and many of them go from one round of financing to another. So it becomes very tricky sometimes also to have this kind of investments done early on, where you already invest into automation at the preclinical stage, or when you're about to file for IND. So, but there's obviously, I mean, if you don't do it, obviously then it creates complications along the way, because as also discussed from the regulatory point of view, I mean, if you were to change your process and add the automation step, then maybe it's not even the original process there, and then you end up with a stability study and bridging studies, and then it becomes time consuming and expensive. So, and I think this is where the tricky part is, and there's a fine line on how to find the right balance between the two, and I think, I mean, as also we see where the industry is moving, with kind of where already phase 1-2 becomes positioned as a pivotal study, then it becomes really critical to move to automation already at phase 1 level. So that to avoid commercialization delays. And we, for example, in AGC Biologics, as the contract and development manufacturer organization, automation is critical to us, and we've been already working on it, on how to ensure that we also make life of our customers easier, and we've been already introducing a lot of automation, particularly for QC analytics and analytics areas, and also on fill and finish, because we should not forget also not just the initial step, but also in the final step.
Elena, you want to add something?
Yeah, so I think all of the colleagues already agreed that you should think about automation as early on. But I think thinking about automation and integrating automation, these are two different things. And I think, of course, in the beginning, in the preclinical stages, you want to remain a certain degree of flexibility. So I think what's important is that when you develop the processes, you think about how the technologies or how for example, the cultivation systems, how they can later scale and translate towards automation. And that doesn't necessarily mean that you have to employ fully automated systems early on. But of course, if you can implement these kind of technologies, then automation can benefit in terms of integrating analytics and collecting a broad set of data in order to generate a good process know-how, which then later can help you with the translation of the processes towards the automation. But I think really the point is thinking about the automation and thinking how the process can scale, even if you're not able to integrate automation at the very early stages. At least pay some thoughts on how you will later translate these.
No, very good points, Ellen. I think you summarized it pretty well, that you should consider automation very early, but keep in mind that I think all the panelists touch on this, that your product shouldn't get impact. And Amina pointed out really well, if you go fast too early and you automate the process and your product like CAR-T, the persistence is the key. If the persistence is not there, you have to go back and make sure that whatever changes you make, how did it impact? So understanding the product, making sure you think about automation early and have that flexibility. So I'm going to go, I have a lot of panelists who want to chime in. So I'm going to, with that mind and with those points, I'm going to go to Stefano and then Deanna on what they have to say, and come back to you, Elvin and Donna, on you started the discussion. So what are your thoughts, Stefano and Deanna? And I will also touch on Fabio before going to you, Elvin and Donna. So.
Yeah, I will be very brief, as most of the points have been already well touched, well presented. I think one aspect that someone already mentioned is also to really take a step back and give a big picture. And to give a real life example, it did happen to me to work with a company where they knew at the end of the day, when they will get to commercial, and they're not commercial yet, their target population is about 5 to 15 patients a year. It's a rare condition. So they decided that in that context, it wouldn't make any sense for them to automate. It would be more of an effort. So, I mean, this is really an extreme situation, but I do think automation is indeed most of the time is a must, and we should be careful indeed of when to start to think of that. But I thought it was interesting also to bring up a scenario that might not be that rare, where perhaps automation will not be of such a benefit.
Thanks. I would like just to add up that exactly the timing is very crucial. What we learned by doing is that it's better to do also your CMC gap analysis rather in advance before going into clinics and starting all the work, because afterwards you're already there and they expect you to have soon a design freeze, which you can compare with the automated version. And then the question whether you compare the different step of your process, which you automate separately, or you have a big machine doing from A to Z, is a totally different story. And from a perspective of finding mistakes or differences at which point they happen is also different. So maybe also modular automation would decrease the risk going towards full automation or exactly the semi-automation. And another point to add is what we see that then the disposables of these automated systems, their pricing doesn't add up so much to the price that you reduce by not doing it manually. At least in our case, it's not a huge difference.
I think that's a really good point, the pricing and modularity of automation. I think it fits right into Fabio's comment when we talk about pricing. So Fabio, what are your thoughts?
Sorry, I was on mute again. So I think I really like the points that have been all covered. Yet it is important to consider all the cost component. And at the end of the day, it's also important to consider the end goal and the patient population. So the size of the patient population, pretty much what Stefan already said. There will be cases, maybe might be rare, but there will be cases where automation is not worth doing it. But that's the key point. And that's why for all the other cases where automation is a must, I would actually stretch automation even before getting into clinical, because you want to look at the size of the patient population. If you're looking at hundreds of thousands of patients, you need to consider that upfront. And there is no way you're going to do that with a manual process. So even though investors might not like spending lots of money up front before even getting into the clinic, I think if you don't do that, especially for a big indication, then it will backfire, because what you want is develop something and then a big pharma is going to buy, because the end goal is to reach to patient, not necessarily to push to phase 2 and then having to redo the development from scratch. So I would say end goal has to be what really drives the decision as to when you automate. And then, as you touched a very good point there on modularity, we have manufacturing processes that change, like they keep changing, and like we talk about gene transfer, and then there are lots of non-viral CAR-T. But then at the same time, maybe in 10 years, people are not going to use gene transfer anymore. They're going to move to gene editing. So the only way you can develop your process then is using something which is, yes, automated, but is also modular. And modular and flexible enough to allow you to change, if you have to, the process later on, but in a structured way without having to start everything from scratch. And I think that's important. So I think alongside the when to automate and why automate at a certain development stage, we should probably add how you automate, which I think is very important, and actually would be interested in listening to the other fellow panel on that.
Yeah, and that's a really good discussion. So I'm gonna take Elvin, Donna, and Tatiana's and Katie's. I think hands are raised. So as we have talked about that, we understand that automation early is good. Take consideration of your patient population. Think about, is it cost benefit to you? So I'm going to elaborate, like as we go through this discussion, and I know we only have an hour, so I'm going to add a question on top of it. Keep those thoughts in mind. And then when you think about various stages, right, we talked about various stages, whether it's preclinical, clinical, but if you think from a process perspective also, is it also good to automate one part of the process earlier compared to another part, right? So if you say, I want to automate cell expansion, I want to automate my cell isolation, but I can think about other parameters of automation. Let's say analysis of automation can come later at stage because I'm still characterizing, I can live manual there. So not only the stages of the clinical development, but stages of the process. So I'll start with Donna and Oliver and move to Tatiana and Katie. Katie, sorry.
I wanted to add on a little bit about what Amina said, that from a regulatory perspective, you have to be really conscientious and understand your process in its entirety. I think there's a lot of tendency to focus on the product quality attributes, the process parameters, and it's tends to be a lot of quality jargon that gets pushed around, and people focus on the product and what the end product is about, its full characterization, potential potency, functionality, without fully integrating that same thought process all the way from the beginning, i.e. the seed material, either your leukapheresis or your cord blood, whatever you're starting out as your seed material. It's just as important to understand the characterizations of that product and how it contributes to your process, and understand that migrating to automation is not black and white. It's quite complicated and complex. You may have a process where you hit the culture phase of your product and it's worked out in a manual system that you activate, then you do gene transfer over X number of hours, and then you do expansion. But when you get into different automated systems, you may need to readjust that process. But what becomes important is understanding what is happening at each stage of that process about your product, what's making it unique at that point in time, and how do you translate that over when you go to automation. I think that's a huge failure. I've seen several companies that, okay, I've got this process, let me put it in an automated system, and it crashes and burns. You don't come out with the same thing. And she gave a good example. You lose your persistence. That's what our products are all about right now for the immunotherapies. So I think there's a tendency to not put enough emphasis on that characterization and understanding your process and what goes on from the very beginning, starting even with your seed material in every phase of your process. What impact is it having on your product, including your raw materials, your media, your activator choice? There's just a lot of components to that need to be understood intensely.
Oliver, you want to go next?
Yeah, I totally agree with Donna. Interestingly, many things we say now for cell and gene therapy also apply to biologics in general, be it antibodies or be it vaccines. The logic is more or less the same. What we've seen is, or what we've heard a lot is, the product is the process. If you change the process, especially in biologics, you may get a different product. So they really have to be developed together. But the counterpart of that is that very often we heard people that say, we suffered a lot to do the process. We receive less attention than analytics, than people working on other parameters. And we ended up doing something for the clinic that was just good enough. And then we had no credit, no whatever to improve the process. So the process was frozen. And as Amina and Over said, once it's frozen, it's very difficult to change, very costly. You're not even sure sometimes what you get if you're changing the process. But then you go for the rest of your life, or the life of a product, with something which is sub-optimized. So when you had 10 CAR-Ts in study, that didn't matter because the price didn't matter. Now that you have hundreds of CAR-Ts coming to the market, you have hundreds of stem cell companies. The one we are helping in Europe is doing stem cell for cosmetic surgery and reconstruction. It's extremely competitive. So they are going to have to master the cost. If you start with a rushed, sub-optimized process, you may have to live with it for a long time.
Thank you, Oliver. So I'll go to Tatiana. And Tatiana, you provided a good introduction that you have a huge experience coming from UPenn to Novartis and the process transfer. So I'll ask the same question. Definitely, when to automate? It's an important question. How to automate? And when you think about stages of the process, when you make a roadmap, do you think some process should be automated first compared to others?
So yeah, I believe that before we automate, we need to have some understanding of the product qualities and the process parameters. And then we actually start looking at the landscape, what's available on the market, what the vendors available, and how do I want to actually automate? Do I want to go with multiple vendors? Do I want to go with one vendor? And basically automation is also choosing a partner. And that's a very serious decision because you want to know that this partner will be with you. This vendor will be on the market for the time of your production, that this partner will actually work with you if your process changes. So if the partner changes their product, so that they will work with you. So I think it's really important when you choose the automation type, you also choose the right partner for you to be with you for the duration of your actually production.
Thank you. So again, do you think when you were developing the roadmap, do you think a particular process should be automated first, or you looked at the process as a whole from end to end?
So I will probably, currently we have options to actually use the automated system from beginning to end, and options to actually automate particular steps. So it depends on the process. So for some product, I would recommend just use one automation system for easiness, but for some, you probably combine different types of automations that actually gives you more control on the steps. And sometimes the wholly automated system doesn't provide maybe the doses. When you have the large dose, you might actually want to go outside of the currently available automation options. So it pretty much depends on the type of process product that you're dealing with to recommend what type of automation and the automation strategy to actually go forward.
That's a good point. So I'll come to you, Elvin. I'll just touch base with Kathy. So Kathy, that's a very interesting point what Tatiana brought, and you're developing solutions. So as Tatiana mentioned that when you are developing automation, you're looking for long-term partners. So what are the challenges coming as a startup scale when you're coming with automation? What are the challenges you think you face? And also your point on the process, which process should be automated earlier than others? So please.
Yeah, lots of good questions in there. I mean, it is a challenge. I think the other piece about being a smaller company kind of coming into this industry, but I think there's also a lot of opportunity because this industry has been working on adaptive technologies coming out of bioprocess or coming out of blood banking. And there's been this constant beat of we need purpose-built systems to meet cell therapy of where it's at. And so I think there needs to be a space for evaluation of these other technologies that are kind of a little bit more built to help, for us, it's the small sample sizes. We can take patient samples into our bioreactor and you can start to use actually sick patient cells to kind of look at your product profile as well. So I think the challenge is as we've all talked about, automate as early as possible. It's how do you consider integrating of new technologies that the industry's been asking for, but now you're already into a phase one and you've automated? How do you consider to, adopt or bring those technologies in? What do tool providers need to provide to allow that consideration to happen, as well as kind of, what is that evaluation that takes place? And you had a second question there. I totally spaced out on it. What was the other one? Oh, on the whether piece by piece or the whole process. I actually agree with most of the people on this panel in that it's probably to change out or evaluate on a modular basis is actually probably the easiest way to do. This market's moving 100 miles an hour. We're building the plane while flying it. All of those little speech talks that we all give. But I think, if you try to fix the whole problem versus each piece, I've always been kind of more of automating each modular, but finding a way to ease the connections between all systems is the better way to go.
Thank you, Kathy. And this is normally how you think scientifically. You want to go from a process changing piece by piece. So I'll go to Elvin and then Zelena. They want to put their comments in. So Elvin, please.
Yes, I would like to touch a few points. So automation also, you need to consider how you design your facilities on a risk base as well as your process, right? So your facility need to support your processes, including automation. So it's funny, in the single-use world, so I used to call like the equipment is on wheels. So you need to have your ability to adapt yourself real quick to different processes as a CDMO, right? That's one point. So going back to your question, what would be the first process that I will automate it? Yes, I think you're right, and that's how we did it in the Fujifilm Diocese in Texas. So we start for the cell expansion. So we actually are currently all the cell expansion to from the incubators all the way up to the 2,000-liter bioreactor to multiple trains that we have in multiple facilities. So all of those are integrated base as a unit of operation in our DCS system. So I'm a big believer on centralized systems. Lucky for me, we have more than 30 automation engineers in our network, and we are capable to support all our partners right now. We like to protect a lot of our intellectual properties and our configuration-based, automation, and for our partners as well. So to give you an example, over the last, I would say maybe 30 months, we have created more than 75 different type of recipes for different processes and different partners out there. and we have been doing that internally. So we create the platform, we build the platform, we have been streaming our processes on how to do this quickly and easier for our customers. And we basically are using the same platform from early stage to late stage, even commercialization. So I think, like Kathy mentioned, so we're 100 miles an hour on this industry, and equipment is changing a lot. Technology is key and it's changing a lot. So we need to take that into consideration. But I think we have a right path now in Fujifilm to do it, and we're moving now to the downstream equipment. That's our second stage that we're trying to get up, and I hope in the next few years, finally vendors and from automation and equipment can talk to each other and they can close that gap, right? There's a big gap. I mean, you have multiple vendors doing automation by themselves and equipment by themselves, but nobody's talking to each other, and we're suffering that in some way. So we need to step in and say, okay, this is what we need. I mean, we are more than capable to do it, and we need to partnership with all of the companies out there to make this happen. So I'm seeing some processes out there right now, multiple vendors talking to each other, to provide the industry what we need, and hopefully that can continue. So,
thank you, Elvin. And with that topic, I will actually go to Zelina, because Zelina talks about automation of the whole process, including robotics. So when we think about modular automation and integrated automation, we touch 2 points. You can have every process integrated into one system, or you can have modular systems integrated through robotics. So what are your thoughts, Zelina, on this? I know you raised your hand, so I'm very keen on learning from you. How do you think those automation technologies work together to take this field forward?
Yeah. So obviously, I'm a strong advocate of modularity as we build more modular systems, as you mentioned, combined with robots, etc. And I think as a production technology institute, we see other industries which are already heavily automated. And the question that we always ask ourselves is, why can't we not automate also the sector of advanced therapy production? Why can't we automate it in a similar to a similar degree? And I think, of course, we have a different history. We have completely different requirements. But still, I think modularity has big advantages. And for these kind of integrated systems, in the first moment, they seem tempting because you get everything out of one hand, and especially for companies that have not so much a background in engineering, for example, they have a solution which is ready and it's fully integrated. But I think in terms of not only in terms of flexibility, I think Alvin and I think Katie have already mentioned new technologies are emerging all the time, and we want to be able to integrate them flexibly. But also in terms of efficient use of equipment. So if you have a fully integrated system which is capable of all these nice steps, for example, isolation, purification of cells, when your process is taking 7 days, 14 days, that's equipment that you can't use because it's busy incubating. The question is that efficient usage of equipment? So yeah, I'm an advocate for modularity. However, I think we have to be careful because there still are a couple of pitfalls, and I think Oliver has mentioned before the topic of interfaces, and I think that's very important, both on the hardware side. So, for example, what kind of disposables are interfacing to each other? How can we interface the different steps? But also on the software side, so we need to ensure that I have all the pieces of equipment that they are able of talking to each other, and that the data that is generated can actually be used, and that the interfaces that are provided, which can vary from vendor to vendor, that they all tie into each other. So I think these are considerations that you have to make when you think about modular automation.
Thank you, Zelina. So I'm going to go to Ramin, but I want to add another question on top of it because of the limited time. So please put your thoughts in. But one question I do have as we go forward, and then I'll come to Amina also. So does that modularity or the integrated approach plays a role when you're thinking about commercialization of your therapies, whether you're going to have a centralized model or a decentralized model? Because as therapy becomes commercial and global, you want to go through decentralized model, maybe not totally decentralized next to the patient, but at least regional decentralization. So when you're thinking about this modularity and integration, do you think that that plays a role of what your financial model looks like in the future? So coming to Ramin and then Amina. And if you have other thoughts, please chime in and we can pass this question later. So
Sure. I think a lot depends obviously on the type of process. Obviously, we're talking about allogeneic cell therapy, or we're talking about viral vector. And I mean, being close to patient maybe is not really an issue, and then process is already more scalable. And it also depends if your starting material is, for example, is fresh and it's fresh in and fresh out, or it can also be cryopreserved, because then again, I mean, every process is different, obviously. So, and I think that also has a massive impact on how to make a therapy commercially viable, and how to ensure that it's actually available to patients, because I would imagine, I mean, for certain diseases and for certain processes as well, it would not make sense. Decentralized approach would not make sense, and I think the key point, what we also need to keep in mind, and this is also tied to centralized versus decentralized model, is on the volume of patients we're talking here. And it has already been mentioned in these discussions that the industry is moving at 100 miles an hour, which is true. But we also know what we need to add is, so far as the approvals that took place for the products which we now have on the market, it's still for not for very, I mean, we're still talking about treating thousands of patients. So even with the CAR Ts approved, I mean, their estimated range is more in thousands. It's not in tens of thousands or hundreds of thousands if the approval were to made for a common indication, like diabetes or some major cardiovascular diseases. And I think it's a bit of a chicken or the egg here, because on the one hand, we need those kind of an automation and automated solutions to ensure scalability. But on the other hand, I think the industry can still take this semi-automated processes and still make it viable, because we're not talking about treating tens of thousands or hundreds of thousands of patients yet.
Amina, you want to add your comments and then I can move.
Yeah, I'm just going back to sort of what everybody was talking about with the industry moving at 100 miles an hour. I think a lot of the time, sometimes we forget about what the regulatory requirements are, because those are moving very, very fast as well. And the goalposts are changing very, very quickly, because the MHRA and the EMA are still learning as they go along as well as these products develop. So they're constantly moving towards standardizing all of these products, standardizing all the specifications, because at the moment there are no specific specifications, and there's massive variations from one vendor to another, from one manufacturer to another. So as we move towards standardization, lots more specifications are emerging, a lot more different requirements are coming through from a regulatory perspective. So moving really fast with the technology and with the automation might actually end up clashing with the regulation, depending on what state is emerging. So it's quite it's well worth keeping an eye on that, really. So that I just wanted to mention that.
No, that's very good. And I'm going to go to Donna and Oliver after that. You actually improved my question a little bit more. So my question was, definitely we all are kind of agreeing that modular approach makes sense. It will definitely depend on your product and the process. But when you think of modular or integrated approaches to be taken for automation and thinking about your centralized or decentralized model, also the regulatory impact is there. So Donna, what are your thoughts? I think you added a really good flavor on the question, which I was not thinking about. So thank you.
I just wanted to make a comment about the centralized versus decentralized. As a company, you own that product. And until you really understand all the nuances of the characterization, the potency, and how it really impacts patients in the clinical setting and across various disease entities, you may have a platform technology, but you don't have any guarantee that that process is going to work the same if you're working with a glioblastoma versus liver cancer versus GI disorders. So I think, there's a lot of thought about that, and decentralized has become a buzzword out there. But as a company, I own that product and I've got to have control of it until I completely understand all the nuances, what impacts it. There may be a point in time in the far future, much more close to biological launch than early stage, that decentralized may play a role. But then, it's right now the default is, well, there's a lot of cell and gene therapy academic centers that can do these processes. Yes, there is, but you have no control that is being done as precisely to the law, to the, dot the I's, cross the T's, the same as you are in your manufacturing environment. And it's challenging enough to assure that as you work with CDMOs, that there's no loss in that technology transfer. Even as you go through, you may start out with the CDMO with, here's my base process, but I am improving that process. And assuring that you translate all of your process improvements and migration to automation effectively with the CDMO is challenging enough, let alone thinking about trying to decentralize early on.
So we looked at this topic for a European company a few months ago that wanted to come to the US and started to ask, do we need 1 center, 3, 5, 20 centers in the US for stem cell therapy? And we came out with 4 considerations. One is economies of scale. And we are back, I think Elvin mentioned it, in terms of designing a process and making sure that, that you have the right process with the right scale that was going to centralization. Number 2 was quality. And in terms of quality, collecting data, understanding the data, what may go wrong, I lose this batch, why do I lose this batch? And having the right people to look into it and not creating the complexity of 10 different centers that went to centralization. Number 3 was regulatory, went to centralization. So number 4, which was the last one, was really logistics. And this is a procedure that needs to obviously take cells from the patient, work on them, and then send them back to the patients. So we looked at different locations, and just to illustrate, but we looked at a location called Lake Nona Medical Center, which is adjacent to the Orlando airport, which I think is one of the airports with the most traffic in the US. So you can actually either fly a patient or fly a sample to this place, which is attached to the airport actually, and there's thousands of or dozens of square miles available for doing this kind of work. And then logistics is not simple, but it's solvable with the right partners, and all the other elements were going towards centralization.
Thank you, Oliver. And I would ask Tatiana now. So Donna touched a really good point of technology transfer to CDMO, and decentralization may be too far unless you understand the process and understand how to control it. So I'm going to ask Tatiana, and then I'm going to move to Stefano. So Tatiana, what are your thoughts on these considerations and modular approaches integrating into that?
So yeah, decentralization actually made it possible, automation made it possible to decentralize cell therapy production and put it closer to the bedside pretty much. And so there's like 2 different options, cell therapy hub versus bedside. But technically, when even the bedside trials decentralized, they move forward to late stage, there is a tendency to put all those like multiple automation devices in one place in cell therapy hub, and it can be CMO or it can be actually the someone's manufacturing facility, so that there is a better control on the quality, on the release criteria, and the raw material control, so that actually at the late stage, especially commercial, we need actually to have a better control on what's we actually putting into the process and what's the actually is getting out of the process. That's why the centralization might work for the early stages, but then at least currently there is a tendency to actually put the devices back into the hub, and it can be both CMO or just someone's manufacturing facility.
Thank you. Stefano?
Again, there's been a lot of very interesting comments, and the only point I think I would like to add, and perhaps I'm trying to address a little bit your question, Dalip, is going decentralized, and I've been exposed to a couple of projects on that topic, add complexity. I mean, that's a fact, and I think everyone agree. Personally, I'm a big fan of modularity from a process perspective. However, I personally think that if decentralization exists, the more you decentralize, meaning the more you get to a point where the hospital is actually the manufacturing hub, the more you will have to look into an integrated solution, where you have to get to a point where basically there is something goes into a box, you push a button, and then you get things out. And then, of course, there might be different way to communicate to the owner of the product a certain amount of information so they can keep track of what's going on. But the more you decentralize, the more I feel it's necessary really to get away from modular, have just the box, and let it go.
No, that's a good point, and I totally get the total decentralization next to the patient compared to a regional decentralization where you make a new hub, but not sending all the samples from all the globe to US, for example, and sending them back. So last question, and that's a really good discussion, and I'm sure I can discuss 4 to 8 hours with this panel. I have so many questions. Just to touch lastly, if we talk about one step, the cell expansion step, if any one of you want to share your experience that how does that cell expansion step, automation can help in that cell expansion step, not the whole manufacturing, but on that cell expansion step, if you can highlight automation helping you there, or increasing your process efficiency, or helping you transfer your manual process to much more higher yield. So I see Elvin.
Yes, I mean, if we're going to talk about cell expansion, so I will say maybe integration with the operator on all the phases and the steps, right? So we talk about if we're going to move to commercialization on that product, so you're going to repeat the same process. It's like cooking, right? So you need to do the same steps at the same time. So you guarantee the quality of the product you're putting out of the patient. So you increase quality and you decrease your risk, right? Or even losing a batch or your processes. So an interesting point going down to that workflows. So most of the industry, especially in the early stage, are paper-based, right? So when you're talking about batch records and documentation, and we found a way to integrate workflows into the recipes, so we can guarantee that things happen at the point that we need them to happen, because scientists already put their input and customers put the input into that process, and we need to make sure that things happen in the right time. So we can repeat over and over and over. It's like same thing with the data. So having centralized data, you can use that centralized data not just to store the data, right? If you start using the data, so you can analyze how you can improve and get better processes in place, and how you can get even things quickly during your fermentation process or cell expansion process, right? So I think automation play a key element on that one, especially on the late stage and commercial stage, right? So we're talking about different steps and different requirements for different automation processes depending on where you are in your process, right? So
sorry, Elvin, and sorry everyone. I just getting a text message that it's only 1 minute or 30 seconds left. I would love to hear all of you from all of you on the cell expansion, but I would like to thank you all for coming to the panel. I cannot thank you enough. It's such a diverse panel, as I said before. I'm so happy to discuss for 8 hours with this panel, but I only had 1 hour. But thank you again so much. Have a great day and have a great conference. And sorry if I could not take your comments or things if I missed things. So please, my apologies for that. Have a great day. Thank you so much.
Thank you.